Yeah, so thanks again for having me. It's my great pleasure to be here. Um, as Julia mentioned, my background is that I'm a physician. I work in internal medicine in a hospital here in Melbourne, Australia. And my PhD was in infectious disease ethics, and much of it was actually focused on the project that became this book. Um, and I've just started at the University of Oxford Wellcome Institute for Ethics and the Humanities, although remotely, since I'm not allowed to leave Australia at the moment for pandemic reasons. This work was funded by the Wellcome Trust, and I've also received funding from Brochet and WHO, but I don't have any financial interests in any of the things we'll discuss today. I've never participated in a human challenge study, but I would really like to. The only place we do them in Australia so far is in the far north of Australia, about 3,000 kilometres from here, but they're talking about setting up a challenge centre in Melbourne. I'm 36 years old, so I would be too old for the first round of coronavirus challenge studies if they went ahead. I would personally be willing to participate in one, but I've been part of groups that said that at least the initial study should focus on even lower risk groups. So that's some background about me. Today, I'm going to mainly present the findings from this project that was published in this book with a terrible academic book cover there, Human Challenge Studies in Endemic Settings. I'm going to give some background of that project, talk about our conception of what human challenge studies are, some of the historical cases, some ethical issues that we identified, and then at the end, I'll talk about some implications for COVID-19 challenge studies. So the Wellcome Trust, who is a funder of other types of challenge studies, issued this call in 2017 that this project was formed in response to. They asked us to review existing challenge studies or controlled human infection studies in endemic settings, meaning predominantly low and middle income countries, to inform an international ethics framework. And this project was led by Professor Michael Selgalid, who was my boss at Monash. And we also had some assistance from Jane Brophy, a social scientist, as well as some great experts that we interviewed. The project involved an extensive literature review looking at the history, ethics, and regulation of challenge studies, some qualitative interviews, and then conceptual, qualitative, and ethical analysis. We spoke with 45 experts all around the world, working in just about every continent. Predominantly, we spoke with scientists who had expertise in challenge studies or had conducted challenge studies, as well as ethicists with relevant expertise, regulators, and funders. So one of the first questions in any area can be, questions about definitions. What do we mean when we say human challenge studies or controlled human infection studies? And when we set out to do this project, there wasn't really a clear definition of what really counts to be called a human infection challenge study. So we suggested that it involves these three things. Challenge studies involve intentional infection with the aim to test vaccines, study infection or immunity, or develop models of infection so as to do those first two aims. And it involves doing that in a highly controlled setting. This is probably old news to many of you one day sooner volunteers. But an interesting way of thinking about this is that one reason why they're called controlled human infection studies is because there's all kinds of controls involved. So I think David was asking the question about treatment, and there's been a lot of focus on the availability and the goals of treatment in challenge studies. And actually we have a paper coming out that talks about that in more detail. But in general, challenge studies can involve controlling the pathogen, so determining which strain we use, how it's given, how much is given, and when it's given to participants, controlling the environment in which infection occurs, including by providing healthcare support to reduce risk to volunteers, and having good infection control to prevent the spread of infection to others. It obviously involves the control of infection through treatments, whether that's curative treatment or supportive treatment. And it involves controlling the population who's infected. And this is, You know, in the case of COVID-19, for example, one of the most powerful ways of reducing risk is by selecting the people at lower risk. And these, of course, have ethical implications because depending on how well these controls are done, the risks to participants and third parties might vary widely. And the other thing that might vary is the generalizability of results. So if we want to do any type of research into an infectious disease to learn about how to prevent and treat it, well, those results of that study have to be generalizable to the real world in some way. So this leads to trade-offs, for example. It's been proposed, or in some cases done in challenge studies, to use a lower risk strain in the study, for example, but then that raises questions about how can that tell us about infection with the so-called wild type strains that are out there circulating in the community. Likewise, some people have raised questions about if we enroll low risk participants, how can that tell us about high risk individuals in the community? And I'll talk a bit more about that later on. And so it's important to note that challenge studies aren't kind of out there in a vacuum. They share some similarities with other types of research and some important differences. Many people, when they hear about human infection challenge studies for the first time, as I'm sure you know, focus on the intentional infection aspect. And maybe they think about some of the unethical historical cases, such as intentional infection research that occurred during World War II. Perhaps they think about people who deliberately get infected, or you know, types of historical or recently proposed infection control or public health control measures that involve infecting people. But of course, there were also cases of, for example, the Walter Reed yellow fever challenge studies, which were like modern challenge studies, except they weren't especially well controlled. They involved infecting people with the wild type of yellow fever virus. And although they involved treatment, they led in some cases to quite severe outcomes. There's a few types of niche research where people are investigating the therapeutic benefits of infection, and that's a kind of overlap between challenge studies and other types of research. And clearly, there's some overlap between vaccine field trials where people are given a vaccine and then allowed to be infected in the community at random. And in some cases, there's overlap with phase 1 drug trials or first in human trials, which are highly controlled studies using highly controlled populations and so on. You know, there are other ways of studying infection, of course, that don't involve intentional infection and that don't involve highly controlled studies, such as studies of natural history, although these days we don't need to do very many of those because, at least for most pathogens, natural history is well described. Although, of course, there have been, you know, case series and so on of the natural history of COVID-19 as a novel pathogen, for example. So that's a bit of a conceptual map about how I think about how challenge studies fit in with other things, but it's a bit of a work in progress. And if you have any suggestions, please don't hesitate to email me. One of the kind of most fun parts of writing up this project was reading about the history of challenge studies. And as many of you know, these go back to the 18th century with Edward Jenner, for example, testing the early smallpox vaccines using cowpox. What was interesting about the project is that we were asked to look at challenge studies in low and middle income countries, so-called endemic settings. And if you look back at history, the sort of earliest challenge studies in the 20th century were mainly done in endemic settings. These were studies of yellow fever done in Cuba, for example, malaria challenge studies done in Italy, which was endemic for malaria at the time. Dengue challenge studies done in the Philippines, in the north of Australia, and in other tropical locations, and leishmania challenge studies done in India. And then, of course, moving on in the 20th century, in the middle of the 20th century, there were a number of infamous cases, not only the experiments done during World War II, but sexually transmitted infection studies done in Guatemala, and hepatitis virus infection studies done in the Willowbrook Home for Children. And these cases became paradigm cases of unethical research in research ethics discourse. But it's important to note, of course, that it doesn't necessarily mean that just because research involves intentional infection that it's unethical. I'm sure most of the people in the audience probably realize this. But what many people don't realize is just how many studies have been done. We found at least 40,000 participants had enrolled in challenge studies since World War II. But what was interesting about this is that less than 1% of those had been enrolled in low and income countries, even though almost all of the burden of infectious diseases at a global level occurs in low and middle income countries. And so many might think that there's a kind of issue of justice here, that we need to do more research in the places where and in the populations in whom the worst burden of disease occurs. As many of you know, there were some great early successes of challenge studies, not only the Jenner experiments showing that cowpox prevented smallpox by doing an early smallpox challenge study, but these early studies of vector-borne diseases showed that, for example, yellow fever, malaria, and leishmania were transmitted by mosquitoes and sandflies. And now that we know that, it kind of seems so obvious, but at the time, people had been arguing about how malaria had been transmitted for hundreds, if not thousands of years. And Patrick Manson, who was the sort of top tropical disease expert in the United Kingdom at the time, noted that, at the time, when, by the way, he infected his own son, who was a medical student, with malaria, and then cured him with quinine, many people were surprised that this pathological puzzle, the question of how malaria is transmitted, which people had been debating for centuries, could be demonstrated in such a simple way, just by getting people to be bitten by mosquitoes and show that malaria was transmitted. This was kind of a remarkable scientific advance that changed the way we could act at a public health level to prevent diseases like yellow fever and malaria. So one of the questions in our research at a global level, and one of the reasons I got into infectious disease ethics is that I was interested in global health justice, was asking this question about why have there been so few low and middle income country challenge studies? And when you think about it, many of the kind of reasons that we identified, although it's hard to know which ones are the most important reasons in practice, kind of apply to some of the roadblocks that have been run into during this pandemic. So, for example, some of the reasons include the lack of research infrastructure, healthcare resources, and public health infrastructure, but also some issues about regulations that, because so many vaccines are predominantly developed in wealthy countries, and because populations in low and middle income countries don't want to be the first to be experimented on by something that's developed elsewhere. There's regulations in place to protect them, to require that the vaccine, for example, is tested in a wealthy country before it's tested in an endemic country. But of course, there's also some ethical reasons, maybe, why you might think that at least in the last 50 years up until recently, there haven't been so many low and middle income country challenge studies. For example, some people might be concerned that in populations who are less wealthy, that there might be more worries about exploitation or so-called undue inducement that I'll discuss later on. And some people might also worry that some challenge studies, especially challenge studies that are just investigating the pathophysiology of disease, don't necessarily lead to direct benefits like a vaccine that gets produced as a result of the study. And one idea in international research ethics is that we should only do research in vulnerable populations if there's a benefit that those populations serve to gain from the research. But in the last kind of 10 years, there's been some progress towards a more global base for challenge studies. From 1992 to 2018, there were only about 11 or 12 challenge studies with only 250 people in low and middle income countries. And those were in Thailand, Kenya, Tanzania, Gabon, and Colombia, Latin America. But there's been a number of recently proposed challenge studies at the other places marked on these maps of various other types of pathogens. So it's becoming more of a global network of challenge study researchers. Rather than go through all the ethical issues we identified in the book, I'm just going to talk about a few of them in the talk today. But many people, when they talk about ethics or research ethics, for example, the first thing they think about is risk. But I think it's also important to think about why we do science at all. Ultimately, we do clinical trials and clinical research in general to try and bring about benefits. And in most cases, in most relevant cases, those are public health benefits. And those benefits are what we are weighing against the risks when we try to decide whether the risks of a particular study or set of studies are acceptable. A standard way of thinking about challenge studies is that there is no individual benefit of participation, in the sense that suppose you are in the United States or Europe and you are participating in a malaria challenge study. If you receive a malaria vaccine, that's a very little benefit to you, especially since it's an experimental vaccine and we don't know at the start of the study whether it will benefit a person at all. And you know, in some cases, you might think that getting infected with a pathogen under certain circumstances might at least give you some kind of benefit in terms of immunity. But if you're not going to see that pathogen in daily life, then it's not very beneficial to you. But of course, given that this project was looking at challenge studies in endemic settings where people are actually going to see the pathogen in daily life, there's an argument that developing immunity as a result of being infected in a study might be conceived of as a benefit, although that's quite controversial in the ethics community. Of course, risks are incredibly important, and that's not only risks to participants, which are the main ones I'm going to talk about today, but also risks to third parties. That is the importance of preventing the spread of infection from participants to the wider community. And that's one reason why challenge studies are often done in isolation facilities, at least for certain pathogens. And there's also a set of ethical issues, which I won't focus on so much today, but I'll discuss at the end about the appropriateness of payment, how participants should be selected, and how consent should be conducted. There are and have been enormous public health benefits from challenge studies. As I mentioned, those early challenge studies with vector-borne diseases like malaria really changed the course of public health by showing how they were transmitted. There's now multiple examples of how challenge studies have accelerated vaccine development. And one of the proposed challenge studies is a challenge study in Africa looking at ways of improving the rotavirus vaccine for African populations. So that's a kind of new wave of potential rationales for challenge studies, is about improving existing vaccines. But they can also tell us really important things about immunity and being able to define what it means for someone to be protected against an infection, which is so-called correlates of protection, can be really important for public health. And there's probably also a narrow set of cases in which challenge studies could help us test and refine treatments. Certainly for diseases that are causing a lot of cases, you might think that actually treatment can be done in hospitals. It can be, we can test treatments in people who are infected in the community. But of course, there might be a narrow set of cases in which we could test treatments in challenge studies in various ways. This slide's probably old news to most people who've signed up to One Day Sooner. So just to say that there's multiple ways in which challenge studies might accelerate phase 3 trials. A standard way is that we could select vaccine candidates from among multiple candidates and just get the ones that look the best in a challenge study before testing them in a field trial. That's a standard way of using challenge studies. There's been proposals for emergency use, that is testing a vaccine in a challenge study and then putting it through a safety cohort and rolling it out much more quickly, although that has raised questions in some quarters about how we could monitor the safety and efficacy of the vaccine after rollout. And what's becoming more widely accepted is that once we have a first generation vaccine, say for COVID-19 or for any other pathogen, well, what's really difficult is comparing one vaccine against another. For statistical reasons, that requires a really big population. It's very impractical and it can be hard to justify. So a challenge study could be an extremely powerful way of comparing a first generation vaccine to, say, many of the other candidates, 10 or 20 other potential candidates, to work out if we could find a superior vaccine. And even if we found a vaccine that was 10, 20 or 30% more efficacious than the first generation vaccine, well, that could be an enormous public health benefit in some cases. And presumably many of you can already see the ways in which any of these would save potentially a lot of time. But a kind of less well understood aspect of challenge studies in terms of benefits is how they can help us understand immunity. The correlates of protection that we use in hospitals and in public health to work out whether someone's immune to influenza were tested and verified in challenge studies. When it comes to coronaviruses, almost every useful thing we knew about coronavirus immunity prior to this pandemic had been shown in challenge studies with common cold coronaviruses at the UK Common Cold Unit in the 70s, 80s, and 90s. And this told us really important things about how long people remain immune for, because they did studies where they infected people once and then brought them back in 1 year and tried to reinfect them with the same strain or with slightly different strains. And just about everything that was shown in the Common Cold Unit studies of common cold coronaviruses is being borne out with COVID-19. So this is like an incredibly powerful data set that was developed through challenge studies with related viruses. But looking towards kind of the present and the future, challenge study designs are helping us understand immunity to other respiratory viruses, such as respiratory syncytial virus, which is a particularly dangerous virus for young babies, but also for older people. And there's recently been challenge studies conducted in London, I believe, where they have successfully and safely challenged older people with RSV under controlled circumstances. And it's certainly, challenge studies are certainly helping us understand immunity to malaria. There's some fantastic work being done in Kenya, among other places, that is investigating how it is that adults in sub-Saharan Africa who've been infected with malaria many times can develop a kind of immunity, even though it's not sterilizing immunity. And it's hoped that in the future that might lead us to develop vaccines for malaria, which is one of the oldest diseases of humankind. But another thing that's kind of even less well understood here is that there have been both unexpected benefits and unexpected harms in challenge studies. And I think this reflects that there's kind of more uncertainty about familiar pathogens than we care to admit. These are kind of pretty niche, kind of nerdy findings here, but in the case of Vivax malaria, there was always a question about why some people didn't respond to treatment. And a challenge study was able to show that it was for genetic reasons. Some people responded to the treatment better than others, and that's led to some improvements in the way Vivax malaria is treated. But we didn't know that we would find something like that. The study wasn't designed to find that, actually. That wasn't a kind of unexpected thing that happened in the study that involved both unexpected harm to participants because they got a relapse of malaria, and unexpected benefits because it taught us about how that therapy really works in practice. Challenge studies have taught us about the pathophysiology of dengue virus disease, where it's always been thought among dengue experts that fluid leaks inside the body were a sign of severe dengue, but challenge studies showed that fluid leaks occurred even in asymptomatic dengue, and that's kind of changed the way we think about dengue virus. Worryingly, it also showed us that malaria can cause effects on the heart. There have been at least 2 cases of myocarditis occurring among malaria volunteers, and this is something that we didn't know about until it occurred in challenge studies. It couldn't be featured on the consent form, for example. We didn't know the probability of it, and that's taught us important things, but it also reveals that we weren't as certain about malaria as we thought we were at the start of those studies. Ultimately, ethics requires answering difficult questions about when benefits outweigh risks. And that seems, it's widely agreed that that's more likely in cases where there's strong scientific justification, where it's plausible we'll get some public health benefit, where risks are minimized and where uncertainties are considered acceptable. And ultimately, these decisions require data, in some cases modeling data, and they require ethical analysis. In terms of those risks, without kind of laboring these points too much, clearly participants are at risk of not only acute infection, but that is acute meaning short-term, but in some cases lasting harms, and they're at greater risk if they escape the study and don't receive the kind of monitoring and or treatment that they might otherwise receive. There's potential risks to third parties of transmission, and that's the reason for tight infection control. And there's also risks to research or to the reputation of researchers, because bad outcomes in any type of research, but especially research with healthy volunteers who stand to lose a lot if there's a bad outcome, can undermine confidence in research. And that's one reason why challenge studies, among other types of related research, need to be particularly carefully controlled and conducted. I just wanted to talk briefly about this idea of lasting harm. So a standard view of challenge studies is that they should only involve pathogens that are either treatable and or self-limiting, where self-limiting means that they get better on their own or that they don't lead to lasting harms. But when we reviewed challenge studies, it was clear that many of the pathogens that are used are associated with small risks of lasting harms, including Guillain-Barré syndrome, which is a neurological condition involving paralysis that can occur after infection with a wide range of pathogens, including influenza. Many challenge studies of bowel organisms can be associated with post-infectious arthritis and irritable bowel syndrome that can be long-lasting. And dengue virus, for various reasons, is more severe the second time. And so if you get it for the first time in a challenge study, that might lead to a kind of longer term risk for you. And so these kind of background data can be important for debates about COVID-19, for example, where people have said that it wouldn't be acceptable if there were any lasting harm, because that's never been done before in challenge studies. And I think lasting harms are incredibly important. We need to be upfront about them. We need to be incredibly careful to prevent them. But the existence of lasting harm in itself doesn't necessarily render the study unacceptable to start with, but it is one very important consideration. I'm not going to talk too much about payment, but just to say that it's widely accepted that challenge studies involve a lot of participants' time. People often have to live in kind of quarantine for several weeks. And it's widely accepted that they should be paid appropriately, and that even minimum wage for that amount of time commitment adds up to quite a lot. But it's more controversial to say that payment is designed to be for risk. One reason for this is that what's supposed to make the risk of research participation acceptable, ethically speaking, is the benefits to society that research can achieve, not the payment that the participant receives. Another reason that it's controversial is that some people worry that it could lead participants to take on more risk than they might otherwise accept, and that's so-called cases of undue inducement. But in our project, we identified other potential issues related to payment. Some issues related to over-volunteering, which isn't necessarily a bad thing, but it could be a bad thing if it undermined the science or led to greater risks. There's also the potential for highly paid studies to detract volunteers from less well-paid studies. And so that's one potential kind of outcome at the meta level of research. And some people worry that if we pay volunteers, then scientists might treat them differently, although that's never been sort of shown in an empirical study. That's just a concern people have. Clearly, when it comes to challenge studies, it's incredibly important that participants are carefully selected. And one way of thinking about this is we're trying to strike a balance between achieving generalizable findings that can get us a public health benefit while reducing risk to participants. And sometimes there's a trade-off between those. So for example, in malaria, the greatest harms occur in African children under 5, and no one's suggesting that we should do a challenge study in that group. So we have to find a group who's close enough to the risk group that it can tell us something about them, or that's close enough to the kind of group we're aiming to vaccinate, which might not be the same as the risk group. So many people, for example, have said in challenge studies for COVID-19, how could challenge studies in young, healthy people tell us about the vaccine efficacy in older people? But often these situations are more complicated than we think. So for example, although it might be intuitively plausible, and I think it is plausible, that we should probably do more challenge studies in African populations to understand infection in those populations, Well, just recently, a typhoid vaccine challenge study in UK adults was replicated in Nepalese children. And so it's not always the case that the populations have to be ethnically similar to give us useful results. But importantly, vaccines, the most powerful effect of vaccines is not actually protecting the person who gets vaccinated, but protecting others. And so it might be that we want to do the study not in the people who are at the highest risk, but in the ones we want to vaccinate to protect those people. So I think that raises a whole set of interesting questions. Challenge studies have led to a whole bunch of innovations in consent. This is an extract of the consent form from the Walter Reed yellow fever studies, in which people accepted that they would endanger their lives, but that they would receive great care from the study team, and that they were highly likely to catch yellow fever while they lived in Cuba. And this was one of the very first consent forms ever used formally in research. But challenge studies have also led to a number of other innovations, such as rigorous tests of understanding prior to enrollment, multiple information sessions for participants prior to consideration of enrollment. And there's also been some great social science done, for example, in African challenge studies, showing that even people with very widely varying educational levels can understand information relevant to participation enough to make them able to consent, so long as that information is presented in the right kind of way. So that was a bit of a whirlwind tour through our project, which is a very long book, which I encourage you to download and read. It's open access, and please contact me if you have any comments. But now maybe I'll talk about just some implications for COVID-19. And clearly, some of those issues, like the ones on the screen here that I've mentioned about challenge studies in general, and challenge studies in endemic settings, clearly have implications for COVID-19. Sounds like a cute kid in the background there. But one of the other issues that I haven't brought up because it wasn't part of our original project is a question about deciding whether one type of research is ethically justifiable. Partly depends not only on the study itself, but what the alternatives are. And so just in the next two slides, I want to talk a bit about field trials. And many people have underestimated how difficult field trials are or are going to be for COVID-19, for example. You can't do field trials where there's elimination. So for example, in places like New Zealand and in parts of Australia, there's no coronavirus at all. And so it's impossible to do a field trial of a vaccine in that population. We'll never find out if the vaccine works there because nobody's getting infected. Likewise, where there's lockdown of various levels of stringency, a field trial could take a long period of time. It might take years to get a result in those kinds of populations. And so that leaves us with questions about where and when to conduct field trials, say for COVID-19. And the best case scenario is that we conduct it in the blue part of this graph. When the epidemic is taking off, we get all the people in the population vaccinated, in the research population vaccinated. We let them go out into the population. We work out who gets infected and whether the vaccine is better than placebo. But if we start too late, then it can be impossible to conduct field trials. And if the population goes back into lockdown, into the orange zone, then the field trial effectively stalls. And, you know, the UK is effectively going back into lockdown or threatening to lately, and that might have implications for the trial being conducted there. Another implication is that field trials not only require very large numbers of participants, so many of you will know that the current coronavirus field trials involve between 10 and 30,000 participants, but they also require very large epidemics. So here on this graph, on the x-axis is the number of people in the experimental vaccine group in the study, and on the y-axis is the size of the epidemic in the population in which the field trial is conducted. And the lines represent some real vaccines. So the measles vaccine is the blue line. That's the best case scenario. That's the kind of coronavirus vaccine we really want. But more realistically, the orange and purple lines represent the efficacy of influenza vaccines. And the green line at the top represents the WHO minimum efficacy for coronavirus vaccines, which is 50%. So if you take the Oxford trial, for example, which has been conducted in the UK, during that trial, there needs to be between, let's say, 20,000 and 400,000 cases occurring in the population where the field trial is being conducted in order to tell us, to get us a statistical readout on whether the vaccine actually works. And so field trials really require very big epidemics. And not every population is willing to put up with those epidemics. And some populations have had such large epidemics before that they might not actually get epidemics this large for various reasons. So one question about the ethical acceptability of challenge studies needs to be a comparison between the likelihood that we'll actually get an answer from field trials in a reasonable amount of time. And this is a paper, this is taken from a paper that a colleague of mine and I are going to publish soon, hopefully. So that leaves us with some unresolved questions, and there are lots of them, but I'm just going to highlight three. One is, how do we make trade-offs between these studies of 10,000 people in field trials versus, say, 150 people in a challenge study? And how do we think about the ethical acceptability of exposing 15,000 people to an experimental vaccine as opposed to doing a challenge study? I think there's some clear unresolved questions about the roles of challenge studies in pandemic preparedness. And whether we should be better prepared next time and for what scenarios, just thinking ahead to the future, even beyond this pandemic, because there will be another one. And then there are these questions that I think David raised at the start about, well, how important are various different types of risk minimization strategies? So kind of two of the ones that have been most strongly proposed to justify the risks of challenge studies are, some people think that background risk is very important, and some people think that treatment is very important. With regards to background risk, I actually think this is a bit of a mistake for challenge studies, even though I've written about background risk myself too. It's a mistake for COVID-19 challenge studies. And one reason for that is that risk is made up of probability and magnitude. So how likely you are to get a harm and how bad that harm is going to be. And if you're a young, healthy volunteer in a COVID-19 challenge study, your risk is actually very low. And so whether there's background risk in your community only modifies your risk by a percentage of your background risk. And that actually ends up being a very small percentage compared to some of the other things that we can do to reduce risk. And on that question about treatment, I think David's version of that question is an interesting one. What are these treatment requirements really about? Are they about preventing bad outcomes, or are they about being able to do something proactive? And I think it's not always clear. Some people want one thing, some people want the other thing. And we now have, probably dexamethasone is the best treatment for severe COVID-19. It's by no means a panacea. It's once you get to severe COVID-19, it's a bad place to be, and dexamethasone only saves about 1 in 8 people. But it is a treatment, and moreover, there are kind of other treatments that can be used to reduce harm, even if they're not specific antiviral treatments. But maybe I'll open up to the floor and say I'm happy to take any questions you like, and if we don't get time in the session, please feel free to get in touch. And thanks again for having me.